Hey everyone, Dr. D here, and in this video we are going to cover Chapter 6 from our Hofnagel's Biology Concepts and Investigations textbook. This chapter covers respiration and fermentation, so let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, let's get into it. Chapter 6, Respiration and Fermentation. So this chapter is all about organisms and why we need to eat food. Like this panda bear here, we need to have breakfast, lunch, and dinner each day. We need to consume energy molecules such as sugars and fats, for energy. That's basically what this chapter is about. Now, specifically, we're going to look at the reason we eat sugars and fats. You know how these are energy molecules. You've ever heard that sugars give you energy or that you can get energy from fats, right? This is what we're talking about. And do you guys remember that the mitochondria is the powerhouse of the cell? Do you guys remember that saying? The mitochondria is a organelle inside of our cells where ATP is made. And this is the molecule ATP right here. And its cells use energy in food to make ATP. Molecules in food, such as glucose, this is the glucose you eat in your diet, are used to produce molecules of ATP. And here's ATP right here. This is adenosine triphosphate, ATP. Remember adenosine triphosphate? It basically looks like a nucleotide. It's got a adenosine nitrogenous base over here. It has a ribose sugar. And look, this is why it's called TP, triphosphate. It has 3 phosphate groups attached here. And cells cannot survive without ATP. So it's really important that our mitochondria produce ATP because by cleaving this bond, you see this high energy covalent bond, by breaking this bond and releasing this terminal phosphate, this last phosphate group, we release energy required to do work inside of the cells. So, why do we eat sugars and fats? Is so that our mitochondria can make ATP. And that's what this chapter is all about. How do we make ATP from the sugars and other energy molecules we ingest? Specifically, we're going to focus on sugars mainly. Why does a glucose molecule allow your cells to make ATP? And that ATP then goes on to allow you to move around and do all the work that's necessary to live. And by the way, this process of making ATP from glucose is called cellular respiration. Cellular respiration is the process that makes ATP. All plants and animals and many microbes use aerobic cellular respiration. Aerobic meaning it requires oxygen, cellular respiration being the process of making ATP in the mitochondria. So essentially, the energy from the glucose you eat in your diet is extracted by cellular respiration and put into ATP, and it's the ATP molecule that the cells can directly use. Those cells in your body all require ATP to grow, to divide, to live, to signal. Without ATP, life would be impossible. Essentially, everything your cells want to do that requires energy, usually it's ATP that drives that process and allows that work to be done inside of your cells. And aerobic respiration is a series of chemical reactions. But the overall chemistry of aerobic cellular respiration looks like this. You have the Reactants on the left of the arrow, these are the things required for cellular respiration, aerobic cellular respiration. C6H12O6, which is glucose, plus oxygen gas, O2, are the reactants. These are the things you require for aerobic cellular respiration. And on the right of the arrow, you have the products. So the products that are produced at the end of these series of chemical reactions are CO2, water, and the all-important ATP molecules. And by the way, they haven't written the exact numbers here. So these are not exact numbers of different molecules, but it's showing you the main reactants on the left and the products on the right. And did you know that cellular respiration explains why we breathe? It explains our own respiration. You know how to respire means to breathe, breathing in, exhaling. Well, did you know that the reason you breathe is intimately linked to cellular respiration? In fact, why do we breathe in? What do we want from the air we breathe in? Can you beat Wicket? That's right, Wicket. We need oxygen, right? So look at this jogger here. She's breathing in air, and what she wants from that air is oxygen. And that oxygen is used uh, for cellular respiration to break down glucose. And then we breathe out, right? We breathe out. And what are we breathing out mainly? We're breathing out mainly CO2. Why? Because it is a product of cellular respiration. Isn't that neat? You've been breathing your whole life, right? You've been breathing since you were born. And have you ever wondered why you have to breathe? You know, why do I need to breathe in every day? Why do I need to exhale every day? Why is it bad if I stop breathing? It is simply because of cellular respiration. Isn't that neat? It's because to get ATP, your cells require oxygen. And during that process of cellular respiration, your cells produce CO2, which needs to be cleared out of your body. Isn't that neat? So the reason you breathe is because of cellular respiration. So this leads us to clicker question number 1. Let's try it together. Do plants carry out cellular respiration? A, no. B, no. C, yes. Okay, yes. Remember what it said earlier in this chapter? It said all plants do require cellular respiration. Did you know that it's not just you and me, our cells that have mitochondria? Did you know plant cells also have mitochondria? And that's where cellular respiration takes place. So, it's a common mistake that students make when they say that plants have chloroplasts but animal cells have mitochondria. You see the folly in that statement? It's not and or, it's both, right? So animal cells, your cells, my cells, animal cells possess mitochondria. Plant cells have both, okay? They have mitochondria and chloroplasts. And do you recall? So in the mitochondria, cellular respiration takes place. And do you recall from chapter 5 what takes place in the chloroplasts? Can you beat Wicket? That's right, Wicket. Photosynthesis, right? So plants are special. Plants can undergo photosynthesis utilizing their chloroplasts, and they can also undergo cellular respiration utilizing their mitochondria. Whereas you and I, we don't have chloroplasts, right? So we can only undergo cellular respiration. So let's see. C, yes, they do undergo cellular respiration. They require ATP like other living things, and respiration generates ATP. That's correct. Yes, they require cellular respiration to get rid of extra CO2. No. So the answer is C. Plants carry out cellular respiration, yes, because they have mitochondria. They require ATP like other living things, and respiration generates ATP. So the answer is C. Isn't that neat? So don't forget, plants also have mitochondria. plants also undergo cellular respiration in addition to photosynthesis. Now remember, cellular respiration takes place during a series of different chemical reactions. It doesn't happen all at once. If it happened all at once, too much energy would be released at once, and that would be bad. So instead, we have what are known as the stages of cellular respiration. The first stage is called glycolysis. You may have heard of glycolysis before, and the glycolysis stage occurs in the cytosol, the liquid portion of the cytoplasm. It occurs in the cytoplasm of the cell. Then, if it's a eukaryotic cell, the rest of the stages occur inside of the mitochondria. There is a transition step that occurs inside of the mitochondria. The Krebs cycle occurs inside of the mitochondria. And then you have the electron transport chain, as well as what's known as oxidative phosphorylation, which also occurs in the mitochondria. Isn't that neat? So one of these stages occurs in the cytoplasm, and the rest of these stages occur inside of the mitochondria. So now we're going to get a little bit into the nitty-gritty of the process of cellular respiration. But I want you to know something. This is a biology for non-science majors course, so I'm gonna be a little bit superficial in my explanations. I'm not gonna go into tons and tons of detail, and if I do go into tons of detail, I'll go into that detail, and then at the end, I'll let you know what you need to focus on, because cellular respiration is actually the trickiest chapter in the entire book. This chapter is probably the hardest chapter you're gonna have to digest. So for your benefit, I have a video right above. You can see it right above my head right now. There's a card above me, and that is a link to my For Science Majors cellular respiration talk from the Campbell textbook. If you want to nerd out about cellular respiration and you want to go into infinite detail, watch that video. That's my video where I go into cellular respiration in great detail for my For Science Majors students. So if anything seems like, you know, like it doesn't make sense in this chapter, because I can't go in as much detail with my non-science majors course, if anything's kind of confusing or you wish you had more detail, you could watch that video that I had linked above. Okay, so I hope that helps you, but I'm going to try to explain this as simply as I possibly can. Sometimes it's just really hard because this really is dense material, and sometimes it's not beneficial to explain technical dense material in a superficial way as in a non-science major course. But I'm going to do my best. So let's try to bear with this together. And by the way, again, I'll explain things a little bit more detailed than you need. And then at the end, I'll take a step back and I'll let you know what you need to focus on to do well. Does that make sense? So we can get through this together. So here is a big picture overview of what's happening during glycolysis. Glycolysis means splitting of glucose because glyco means glucose and lysis means to break or to split. During glycolysis, one molecule of glucose, remember glucose is C6H12O6. And here, these gray dots represent the 6 carbons of the glucose molecule. During glycolysis, which occurs in the cytosol, remember in the cytoplasm, glucose is broken in half to produce 2 3-carbon sugars called pyruvate. 2 pyruvates are formed, and in the process, we actually produce a couple of net molecules of ATP. We produce 2 net ATP. We also produce 2 molecules of NADH. And I'm going to explain why this is important later, but what you should understand is during these steps, during these early steps, the glycolysis stage, the transition stage, and the Krebs cycle stage, I want you to realize something. I want you to keep this in mind, okay? Remember that glucose is C6H12O6, okay? During these early stages I just told you about, we are going to be transferring hydrogens. Remember the 12 hydrogens of glucose? They get transferred here to NADH and another molecule called FADH2 later on. Okay, so the H's from the sugar end up on these what are called electron carriers, and each one of these purple dots represents the electrons from each of the H's. Okay, and then the carbons and oxygens carry on in from stage to stage. So again, let's review. During glycolysis, which occurs in the cytosol, the liquid portion of the cytoplasm, glucose is cleaved in half to produce two molecules, two 3-carbon molecules called pyruvate. During this process, 2 ATP are formed, and that's 2 net ATP. I'll explain what that means in a little bit. And 2 NADH are formed because we harvested 2 of those 12 H's from glucose. So that is what happens during glycolysis. This is stage 1 of cellular respiration. Now, after glycolysis, remember we have 2 molecules of pyruvate, these 3-carbon molecules, and those pyruvate molecules are going to move into the mitochondria. So they're going to go from the cytosol into the mitochondria. And during this transition, there is what's known as the transition step. Many textbooks, instead of calling it a transition step, they'll call it oxidation of pyruvate, in case you've ever heard that terminology before. So there's this transition step that occurs between glycolysis and the Krebs cycle. So let's talk about the transition step. During the transition step, remember those 2 pyruvates from glycolysis enter the mitochondria. And when they do, a few things happen, okay? A couple more of the hydrogens are removed from these pyruvates, and those hydrogens are given to NADH. You see, here's one of the electrons from one hydrogen. Here's another electron from the other hydrogen. Those hydrogens are handed to NADH. And this causes the pyruvate to break down. Okay, what that means is that, remember how pyruvate was a 3-carbon molecule? Well, the 3rd carbon pops off and leaves as CO2. And because there were 2 pyruvate molecules, we end up producing 2 molecules of CO2. Do you guys remember, again, do you remember glucose is made up of 3 elements? What are the 3 elements? Can you beat Wicket? That's right, as always, Wicket. Carbon, hydrogen, and oxygen. Okay, glucose is made up of carbon, hydrogen, and oxygen. I want you to understand that glycolysis, the transition step, and Krebs cycle, the main point of these early cycles is to get those hydrogens away from glucose, right? To get the hydrogens away from the carbons and the oxygens. And you see, every time we make an NADH or later on an FADH2, it's because we transferred hydrogens to this electron carrier. And when we take all those H's away from glucose, from the sugar, we are left with nothing but C's and O's. And eventually those C's and O's break down so much that they get released as CO2. Does that make sense? So you can think of CO2 as the oxidized form of glucose. It's the carcass of glucose, right? When we have glucose, we have C's, H's, and O's. When we take those H's away and hand them to NADH, we are left with nothing but C's and O's, which end up leaving our system as CO2. Now, let me ask you this. Okay, let's test your understanding. If glucose had 6 carbons in it, right? 1, 2, 3, 4, 5, 6. If glucose has 6 carbons in it, how many CO2s should we make by the end of cellular respiration? Can you beat my cat, Wicket? That's right, Wicket. 6 carbons in glucose, that means 6 CO2s should be produced. And yes, 2 of them are produced here during the transition step, also usually known as oxidation of pyruvate. 2 of the CO2s are there, so there should be 4 carbons left from our original glucose molecule. I hope that makes sense. Again, please watch my other video. I'll link it again above. I'll link it again above for you. If you want more details, you can watch that video, but I'm trying to keep it more simple for this non-science majors course. So again, what happened? 2 3-carbon pyruvate molecules entered the mitochondria, where they were oxidized to give us 2 molecules of NADH, and 2 CO2s got released, leaving us with 2 molecules, 2 2-carbon molecules. You see this? 2 carbons are left because 1 carbon left. And these new molecules are called acetyl-CoA. We have 2 acetyl-CoAs. And those 2 acetyl-CoAs still have carbon, hydrogen, and oxygen. And those acetyl-CoAs are going to enter what's known as the Krebs cycle. The Krebs cycle, where the rest of the hydrogens are going to be handed to NADH, forming 6 NADH, and the last of the hydrogens are going to be handed to FADH2, forming FADH2, and that leaves us with carbon dioxide, 4 carbon dioxides, as we had mentioned before, and we also produce a couple of bonus ATP right here. So we make a couple ATP. Isn't that neat? So what I need you to understand is, do you see how there's FADH2, NADH, NADH? You see this? All the H's from that original sugar, C6H12O6, all of those H's ended up on these carrier molecules, and all of the C's and the O's left as CO2. That's what you breathe out. You know, when you exhale, you're breathing out CO2. You're breathing out what was left of the glucose molecule. The hydrogens, on the other hand, were handed to these carriers, and you're like, carriers? Where do they carry the hydrogens to? Well, they carry the hydrogens to the very last stage, which involves the electron transport chain, and it's known as oxidative phosphorylation. This is your stage 3. And those hydrogens, the electrons, mainly the electrons from these hydrogens, are what powers stage 3. And that's what gives you the lion's share of the ATP. In this textbook, it says 34 ATP, but many textbooks say 28 ATP. Either way, it's the lion's share of the ATP. Those harvested hydrogens, specifically the electrons of the hydrogens, are what powers this stage 3 to produce the lion's share of ATP for your body. And that's the big picture. And this leads us to clicker question number 2. Let's see if we can solve this together. What happens to glucose's carbon atoms during the process of aerobic respiration? Do you remember? Glucose has 6 carbons, 12 hydrogens, 6 oxygens. And do you remember what I said happens during cellular respiration? We steal all of its hydrogens, and that leaves us with what? Can you beat Wicket? That's right, Wicket. They are released as CO2, right? So we can answer this one easily. It's D. It's released as CO2. We harvest all the hydrogens. We give the hydrogens to the electron carriers called NAD or FAD. Those NADH and FADH2 carry those electrons, those hydrogens, to the final stage, which is called oxidative phosphorylation. And those hydrogens actually power, those electrons power the final stage to give us the lion's share of that ATP. Isn't that neat? And again, where does cellular respiration take place? Do you recall glycolysis occurs in the cytosol, the liquid portion of the cytoplasm outside of those mitochondria? The rest of the reactions, they occur in the in the mitochondria, if it's a eukaryote, all right? If it's a eukaryote, the rest of the reactions occur in the mitochondrion. But if it's a prokaryote, the rest of the reactions continue to occur in the cytosol, because remember, prokaryotes don't have mitochondria, right? Now, if we're talking about eukaryotes, then we have mitochondria. And remember, the mitochondria has a smooth outer membrane and then a convoluted, twisty-turny inner membrane. And do you recall that the folds of the inner membrane are called cristae? Well, there are many enzymes embedded in the inner membrane, and these enzymes catalyze the reactions of the electron transport chain. So you can think of the inner membrane as responsible for stage 3 of cellular respiration. This stage is known as oxidative phosphorylation. It's powered by those NADH and FADH2 molecules from the previous stages. And do you guys recall that the fluid in the center of the mitochondria is called the matrix? So if we take a close look here, take a look. You have the outer membrane that's smooth, the inner membrane that's convoluted. This here is called the intermembrane space. That's the space between the two membranes. And the space in the very center is called the matrix. So you see the fluid in the very center is called the matrix, and the space between the two membranes is called the intermembrane space or the intermembrane compartment. So that leads us to clicker question number 3. Where is the mitochondrial matrix, right? The matrix. Is it outside the outer membrane? No, that's the cytosol. Is it between the inner membrane and the outer membranes? No, that's called the intermembrane space or the intermembrane compartment. Is it inside of the inner membrane? Yes, that's inside of the inner membrane. That's the correct answer. Again, recall that glycolysis occurs in the cytoplasm of the cell. Remember that glycolysis produces 2 molecules of ATP. Remember that glycolysis has inputs and outputs. You should know about the inputs and the outputs. The input is glucose, right? A 6-carbon sugar called glucose. And the outputs are 2 molecules of pyruvate, 2 molecules of the electron carrier NADH, and 2 net ATP. That's what you should know, right? So you don't need to know it in any more detail than that, really. I would want to know, again, for biology for non-science majors, what would I want to know? I would want to know glycolysis occurs in the cytosol or the cytoplasm. Glycolysis requires glucose. Glycolysis results in an output of 2 molecules of pyruvate, 2 NADH, and 2 net ATP. And this leads us to clicker question number 4. Let's answer this together. If 8 glucose molecules enter glycolysis, the net products will be how many pyruvate molecules and how many ATP molecules? Okay, so let's think about this together. Remember that for each glucose, you ended up with 2 pyruvates and 2 net ATP, correct? So if we have 8 glucose, that means we should have what? Can you beat Wicket? If we have 8 glucose molecules, how many pyruvates should we end up with at the end of glycolysis? Can you beat Wicket? That's right, Wicket. 16, right? Because glucose is a 6-carbon molecule, and we're breaking it down into 2 3-carbon molecules. So yes, 16 pyruvates. And how many ATPs? Again, remember, there are 2 ATP per glucose. So again, it should also be 16. So the answer should be 16, 16. 16 pyruvate molecules and 16 ATP molecules for every 8 glucose molecules. Awesome. So I hope you understand glycolysis. Again, I just need you for the non-science majors course to understand the inputs and outputs. Remember, glycolysis occurs in the cytoplasm. It requires a glucose molecule, and it yields 2 pyruvates, 2 NADH, and 2 ATP. Next, remember after glycolysis is the transition step. The transition step is also known as oxidation of pyruvate. And remember, during this step, the 2 pyruvate molecules that form at the end of glycolysis enter the mitochondria. And remember, as they come into the mitochondria, they are oxidized. What does that mean? Hydrogens are removed and given to NAD+ to form 2 molecules of NADH. And remember, those NADH are going to go to the third and final step, oxidative phosphorylation. And during this step, remember, the 3 carbon pyruvate molecules are broken down into, acetyl-CoA. And when they break down, they remove 2 CO2s. 2 CO2s are released. And that's what you exhale. When you exhale, when you breathe out, you're breathing out CO2. And that CO2 is removed from pyruvate. So if you think about it, pyruvate had 3 carbons, 1, 2, 3. The third carbon was removed as CO2. Think about it that way. And now you have a 2 carbon molecule called acetyl-CoA. And you have 2 of them, right? Because you had 2 pyruvates. Now you have 2 acetyl-CoAs. Those acetyl-CoAs enter the Krebs cycle. So that's a lot of details, but what do I need you to know? I need you to know that during the transition step, also known as oxidation of pyruvate, 2 pyruvate enter the mitochondria. Remember, each pyruvate is 3 carbons long. 2 pyruvates enter the mitochondria, and what forms? 2 NADH, 2 CO2, and 2 acetyl-CoA. Acetyl-CoA is a 2 carbon molecule. You have 2 of the original carbons per acetyl-CoA. One of those carbons left as CO2. So you have 2 acetyl-CoA, and those 2 acetyl-CoA enter the next stage called Krebs cycle. Now for the Krebs cycle, remember that at the end of the transition step, we yielded 2 acetyl-CoA molecules. Both of those are headed to the Krebs cycle, which occurs inside of the matrix of the mitochondria. And what I want you to know are the inputs and the outputs of the Krebs cycle. So the main input to know about is, your 2 acetyl-CoAs. These are your 2 acetyl-CoAs. And the outputs you should know about are 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. We're going to look at this in a little bit more detail, but again, you don't need to know all the fine little details. You just need to know the inputs and the outputs of the Krebs cycle, but I'll show you how it works. You see how an acetyl-CoA comes into the Krebs cycle? Okay, when acetyl-CoA comes in, it joins up with oxaloacetate to form citrate. During the subsequent steps, we are taking hydrogens away from the sugar. Remember what we wanted from our glucose is all the hydrogens. Those hydrogens, remember, are transferred to the electron carrier. Here you can see where we've taken the electron carrier NAD+, and we transfer hydrogen to NAD+ to form NADH. And we also result in the release of a CO2. And remember, these C's and these O's were originally on your sugar molecule, and the H's here were originally from the sugar. Remember the big picture? The H's are being transferred to these carriers so that they can carry the H's to the electron transport chain, in stage 3, also known as oxidative phosphorylation. And this causes the C's and the O's to get released as, you know, gas that we, exhale, CO2 that we exhale. Next, we take more H's from the sugar and hand them to another NAD plus electron carrier to form NADH. And again, NADH goes to the electron transport chain to drop off the H, right, to drop off the electrons. And this releases the last CO2 right here. And as an added little bonus, we form some ATP right here. And then later on, we take a couple more H's, and this time we hand the H's to FAD, reducing it to FADH2. There we go. And remember, can you beat Wicket? Where is the FADH2 on its way to? This is the first time we've heard of FADH2, but it's going to go to the same place as NADH. So where is that place? Can you beat Wicket? That's right, Wicket, as always. It's headed to the electron transport chain. Remember, we're harvesting all these little H's from our glucose, C6H12O6. We're harvesting all these little H's during the Krebs cycle and, glycolysis and transition step. We're taking all these little H's because we want to get these H's to the electron transport chain, because that's what's going to power the electron transport chain. And that's why the C's and the O's get reduced as waste, as CO2. Isn't that interesting? So keep the big picture in mind. I don't want you to memorize all these little steps, right? This is a biology for non-science majors course. I'm trying to keep it as straightforward as I can, and it's still going to be hard. I know it's not easy, but I hope you can see the big picture. Next, we have some more chemistry. And look, we transferred the final hydrogens to NAD plus to form NADH, and there you go. At this point, we have harvested all of those H's from glucose, C6H12O6. See, we've transferred them to all these little hydrogens, right, on the electron carriers. And all the C's and the O's, they left through us exhaling. Isn't that neat? So that this whole process, of glycolysis, transition step, and Krebs cycle, this whole process was to get those H's away from glucose, get them on these carriers. So these carriers, NADH and FADH2, can carry those H's to the electron transport chain to power it to make a ton of ATP. And the C's and the O's we really don't have any use for, so that we breathe those out as CO2 gas, right? Isn't that interesting? So again, what do I need you to know about the Krebs cycle? I just want you to know that we had two acetyl-CoAs as input. And by the way, look at this. Just in case you're wondering, look, this Krebs cycle is just showing you what's happening to one of the acetyl-CoAs. See that? One acetyl-CoA. But how many acetyl-CoAs did we have per glucose molecule? Can you beat Wicket? That's right, Wicket. We had two acetyl-CoAs. So what you need to know is that per glucose molecule, you actually get 2 rounds of the Krebs cycle. So you should take all of these numbers and multiply it by 2. So for instance, each cycle produced 2 CO2s. So, and if you have 2 rounds, you're going to have 4 CO2s. You see that right here at the top left? You formed 1 ATP per acetyl-CoA, but you had 2 acetyl-CoAs, so you have 2 ATP. You have 1, 2, 3 NADHs, so you're going to have 6, right? Because you multiply by 2. You formed 1 FADH2, well, you have 2 total, because you have, again, 2 rounds of the Krebs cycle. And that's the Krebs cycle. And guess what? All of these little NADHs, let me show you something. Remember, in glycolysis, you had 2 NADHs that you formed. In the transition step, you had 2 NADHs that formed. In the Krebs cycle, which we just covered, you had 6 NADHs and 2 FADH2s that formed. Well, guess what? All of these NADHs and FADH2s are headed to the electron transport chain. In fact, that's what powers the electron transport chain to give us the rest of the important ATP molecules that we need during cellular respiration. So let me show you how that works. Now for stage 3, the third and final stage of aerobic cellular respiration. Remember, we had 10 NADH molecules that we formed in the previous steps, as well as 2 FADH2s that we formed in the previous steps. In fact, those H's are from glucose. Remember, glucose had C6H12O6. Excuse me. All of those H's are found on here. So you got H's here, H's here. Those H's are now on their way to the electron transport chain, which lives in the Cristae. Do you guys remember the Cristae? The twists and turns of the inner membrane. So what you're looking at here is the inner membrane of the mitochondria. Inside of the inner membrane of the mitochondria, you see how you have these folds that form, these cristae. And in the inner membrane, you have the electron transport chain. And the electron transport chain consists of 1, 2, 3, 4 complexes, protein complexes. Together, these 4 complexes are known as the electron transport chain. And that's where the NADH is coming. Look, NADH comes and drops off its little electrons, right? It drops off the electrons. Because do you guys remember each hydrogen is a proton, which is H plus, and an electron, which is, denoted here as E minus. Each NADH comes along and it drops off 2 electrons into the electron transport chain, and one of those protons pops off here and it floats off into the fluid here, into the matrix. Okay, and so NADH loses H's, which means it becomes oxidized back to NAD+. But you don't need to know all those fine details. But what I would want you to know is that NADH drops off its little electrons at complex 1. This is complex 1 of the electron transport chain. And those electrons go on a journey. See, those electrons go on a spontaneous journey through the electron transport chain from complex 1 to 2 to 3 to 4. And FADH2, on the other hand, look at FADH2. It drops off its electrons, its 2 electrons, at complex 2. And those electrons go on a journey from complex 2, 3, and 4. And so that's actually what powers the electron transport chain. Did you know that by NADH and FADH2 dropping off their electrons, those electrons power the electron transport chain? It's kind of like electricity, right? Electrons flowing through a wire, right? These electrons flow. You see these yellow arrows? These electrons flow, and that actually powers the electron transport chain. So now you might be wondering, what does the electron transport chain do with that power? The power from the electrons flowing through them. Well, look what it's doing here. The electron transport chain, complex 1 is a proton pump. Look, it takes protons from the matrix and actively pumps them into the intermembrane space. Remember the intermembrane compartment or intermembrane space? Well, we're pumping protons into the intermembrane space. Complex 2 is not a pump, but it's part of the wire. Complex 3 pumps protons into the intermembrane space, and complex 4 pumps protons into the intermembrane space. And remember, this is active transport of protons. So we actually end up with a large concentration, a high concentration of protons in the intermembrane space. And look at this. This is so neat, you guys. With that, guess what we do? The protons can then flow out through this enzyme called ATP synthase. Yes, enzymes end in ACE, and this is the enzyme that synthesizes ATP. By having these protons flowing through ATP synthase, a process known as chemiosmosis, you actually are able to make ATP. Look what happens. The power of diffusion of protons, the power of chemiosmosis is what it's called, is what's allowing this enzyme to join ADP, so adenosine diphosphate, with a phosphate group to form ATP, adenosine triphosphate. And you formed ATP, and actually, this textbook says you formed 34 ATP this way through this type of chemiosmotic phosphorylation, it's called. So you make 34 ATP. Now, some textbooks say 28 ATP are made this way, but for whatever reason, this textbook says 34, which I think is an outdated figure, but that's what this book is saying. So again, big picture view, all of those 10 NADH that were formed during glycolysis, the transition step, the Krebs cycle, remember 10 NADH are headed over here. They drop off their electrons. The electrons go on this journey, and that pumps protons from pumps 1, 3, and 4 pump protons into the intermembrane space. FADH2 drops off its electrons. Those electrons join the journey, and that pumps more protons into the intermembrane space. And it's those protons chemiosmosing through ATP synthase that drives you to make the lion's share of the ATP, 34 ATP, according to this textbook. Isn't that neat? So without these H's, right, NADH, FADH2, without these H's, these hydrogens from glucose that you ate in your diet, you would not be able to power these pumps. These pumps wouldn't pump the protons, and you wouldn't make this lion's share of ATP. The mitochondria would truly not be the powerhouse of the cell, right? So, and then here's another thing. This is one more thing to know about aerobic cellular respiration. And that's what happens to these electrons when they get to the end of the ride. Look at this. You see how the electrons are going down the ride, and the electrons get to the end of the ride. So did you know at the end of the ride, something needs to come along and remove the electrons off the ride so that more electrons can come down the ride? Does that make sense? So that something is oxygen. Oxygen, remember how oxygen is a reactant of cellular respiration? Remember how oxygen, you have to breathe in? The whole reason you're breathing in air is to get that oxygen into your blood. The reason is oxygen has a job to do. Oxygen's job is to pick up the electrons at the end of the electron transport chain. You know what they call it? They call it the terminal electron acceptor, right? Because it picks up the electrons at the end of the ride. So it's the terminal electron acceptor. It picks up 2 electrons as well as 2 protons from the matrix to form what? To form water. So oxygen becomes reduced to water. And that's why water is a product. Remember, water is a product of cellular respiration. Oxygen is a reactant of cellular respiration. And now you know why. Isn't that neat? That's the whole reason you breathe, right? Students ask all the time, why do we have to learn about cellular respiration? It seems really cumbersome and tricky. And it's like, it's actually very important, right? It explains why you have to eat sugars and fats in your diet. It explains how you make ATP. It explains why you breathe in oxygen and why you breathe out CO2, right? All of that is explained in cellular respiration. So now you know the whole big picture. Isn't that neat? Now, just to overview, what would I expect you to know about this third and final stage of cellular respiration? I'd want to know that NADH comes along and drops off its electrons at complex 1 of the electron transport chain, and that FADH2 drops off its electrons at complex 2 of the electron transport chain. I'd want to know that oxygen is the terminal or final electron acceptor, and oxygen becomes reduced to water, right? When it picks up those electrons, it becomes water. And I'd want to know that thanks to all the protons that got pumped from the electron transport chain, we formed 34 ATP per glucose molecule right here by this enzyme called ATP synthase through a process, chemiosmotic phosphorylation. Isn't that neat? So again, uh, you don't need to know every little detail, but at least understand the big pictures here. And now let's compare all the processes combined, right? Remember, we had glycolysis, followed by transition step, followed by Krebs cycle, and lastly, we had this chemiosmotic phosphorylation, or again, this third stage is usually called oxidative phosphorylation, but in your textbook, they call it chemiosmotic phosphorylation. Okay, let's just take a look at the big picture. Remember that glycolysis produced 2 ATP, Krebs cycle produced 2 ATP, and lastly, the electron transport chain plus ATP synthase produced 34 ATP. And by the way, I didn't go into detail about this, but the 2 ATP that were made in the previous steps were produced by a process known as substrate-level phosphorylation. I don't really expect you to know what that means, but sometimes your homework assignments and such might ask you. Um, if they do, then the 2 ATP that were made here were made by substrate-level phosphorylation. The 2 ATP that were made here were substrate-level phosphorylation. And then finally, the 34 ATP that were made during stage 3 were made by, remember, chemiosmotic phosphorylation, thanks to ATP synthase. So that sums up to what? 34, let me zoom in here. 34 plus 2 plus 2 is 38 total ATP. However, did you know that it takes 2 ATP to get the pyruvate, or the 2 molecules of pyruvate into the mitochondria? So we subtract that out and we get 36 theoretical yield. We get 36 total ATP here. So what do you need to know? You should know that 36 ATP are produced by cellular respiration. Aerobic cellular respiration produces 36 ATP, and 34 of them were produced during stage 3, chemiosmotic phosphorylation. 2 of them were produced during Krebs cycle, and 2 of them were produced during glycolysis. And by the way, it's not just sugars. Let me show you something. It's not just sugars that feed into cellular respiration to produce ATP. Fats also feed into this cellular respiration pathway and produce ATP, as well as proteins. Proteins can also feed into this pathway to help produce ATP as well. So it's really neat. I know usually when cellular respiration is taught, we're talking about the breakdown of glucose, to produce ATP. But you should be aware that not only does glucose enter the cellular respiration pathway, but fats and proteins can provide ATP as well by feeding into different steps of this pathway. Next, I want to tell you about a similar concept, but a different type of organism. Did you know that there are some organisms that use a molecule other than oxygen as their terminal or final electron acceptor? So you see how you and I, we are aerobic respirators, which means oxygen is our final electron acceptor. It's what picks up the electrons at the end of the electron transport chain and becomes reduced to water. But did you know that some creatures on Earth, not us obviously, but some creatures on Earth, they use something other than oxygen as their final electron acceptor? So they use different molecules. And did you know that's not called aerobic cellular respiration? That's actually called anaerobic cellular respiration. So I need you to know this for the exam. I need you to know that aerobic cellular respirators like you and me, we use oxygen as our final electron acceptor. And you should know that anaerobic cellular respirators, they use a molecule other than oxygen as their terminal electron acceptor. And let me show you some examples of molecules that other creatures can use. And by other creatures, I'm talking about bacteria, I'm talking about archaea, I'm talking about some of these organisms that live where there is no oxygen, right? If you've ever wondered, like, how do creatures live where there is no oxygen? Well, they might breathe something other than oxygen. So let me show you here. Again, you and I, when it says here, what is the terminal electron acceptor? If it's oxygen, that's aerobic cellular respiration. However, look, if it's an inorganic acceptor other than oxygen, for instance, nitrate, NO3 minus, sulfate, SO4 minus, or even CO2 can sometimes serve as the terminal electron acceptor. This is known as what? This is known as anaerobic cellular respiration. Isn't that neat? The only difference is during anaerobic cellular respiration, usually you do not make as many ATP as an aerobic respirator because these aren't as efficient at being terminal electron acceptors as oxygen is. Isn't that neat? So again, if I asked you on the exam, uh, true or false? Humans, true or false? Humans can undergo anaerobic cellular respiration. Can you beat Wicket? That's right, Wicket. No, we can't. Humans are incapable of anaerobic cellular respiration. You know why? Because that would mean we can breathe something other than oxygen, and that's just not possible. So, you and I, other animals, we are aerobic cellular respirators, but there are some creatures on planet Earth that are anaerobic cellular respirators. And remember, let's get Gizmo's help this time. Gizmo, can you tell us again, what is the only difference between anaerobic respiration and aerobic respiration? Can you help us out, Gizmo? Thanks, Gizmo. You see what Gizmo said? Gizmo said anaerobic respiration uses a terminal electron acceptor other than oxygen at the end of the electron transport chain, right? So isn't that neat? So now you know the difference between aerobic cellular respiration and anaerobic cellular respiration. But did you know there's a whole nother type of concept here as well called fermentation? And it's an anaerobic process, but it is not anaerobic respiration, but it is an anaerobic process. So what in the world does that mean? That means that, let me show you something. I'm going to explain fermentation to you next. Now, I want you to think about this. What if you are an aerobic cellular respirator like you and me, and we run low on oxygen, right? So imagine if we are not getting enough oxygen to our muscles. Let's say we're hitting the gym too hard or something like that, and we're just we're burning ATP faster than we can make it. We are not getting enough oxygen. We're breathing really hard. We're trying to get oxygen into our cells, but we're not delivering enough oxygen to our body. Well, do you know what happens? That means we don't have anyone here to pick up the electrons at the end of the ride. And you know what happens? Those electrons back up, kind of like a traffic jam, and this whole electron transport chain shuts down. Isn't that neat? So when there's not enough oxygen, the electron transport chain shuts down when there's not enough oxygen. And also, the Krebs cycle shuts down when there's not enough oxygen. And even the transition step shuts down when there's not enough oxygen. Isn't that neat? So, guess what? When you're hitting the gym too hard and you're panting and trying to get more oxygen into your body, what's happening is your body is only undergoing glycolysis. So you're only making 2 ATP per glucose instead of making the 34 ATP. Remember, or I should say 36 ATP. Remember, you were making 2 ATP here, 2 ATP here, 34 ATP there, for a net of 36 ATP after, deducting 2 ATP for transport. So imagine you're used to getting 36 ATP per glucose molecule via aerobic cellular respiration, but now you're only making 2 ATP. And that's why you start getting cramps. And that's why you have to take a break. If you're trying to work out too hard at the gym, you need to relax and take some deep breaths to get the oxygen back into your body so that you can continue making 36 net ATP instead of just 2. Isn't that neat? That's what's happening when you hit the gym too hard. This is why I can't just go and run a marathon if I've not trained, right? Because my body switches to just making 2 ATP instead of 36. And did you know this is known as fermentation? This is what happens during fermentation. And fermentation is an anaerobic process, but it is not the same thing as anaerobic cellular respiration. Because remember, anaerobic cellular respiration meant that something other than oxygen was serving as the electron acceptor, right? The final electron acceptor. During fermentation, we're just running low on our oxygen, and that's what's causing everything to shut down except for glycolysis. So if I asked you, let me see if you can beat Wicket. Okay, I'm going to ask you a couple of questions about fermentation. Wicket, all right. What causes fermentation? All right. Yes, that not enough oxygen, right? Not delivering enough oxygen to your cells. Now, Wicket. All right, let's see if you can beat Wicket again. Wicket, how many net ATP are produced per glucose molecule during fermentation? That's right, Wicket. Only 2 net ATP, the 2 that are done during glycolysis. And last question for you and for Wicket. Wicket, what is the only stage of respiration that's occurring during fermentation? That's right. Wicket said glycolysis, right? So only glycolysis is going on during fermentation when oxygen levels are low. I want you to know one more thing about fermentation, and that is that different aerobic respirators undergo different types of fermentation and produce different types of fermentation products. So in you and me, I want you to see this. In you and me, as well as let's say these lactobacillus bacteria here, we undergo what's known as lactic acid fermentation. This means that during fermentation, we produce a byproduct called lactic acid or lactate. And so this is known as a fermentation product. Have you ever had a coach, have you ever played sports and you're cramping up and your coach says, take a break, sit on the bench for a little bit, you may have some lactic acid buildup in your muscles. That's what your coach is referring to. You are not getting enough oxygen. Remember, you're not getting enough oxygen. So everything shuts down except glycolysis, which means you're only making 2 ATP. But as a side effect, which I'm not going to go into the great detail here in the non-science majors course, but as a side effect, you're going to make lactic acid. And that's why you get lactic acid buildup during fermentation. Isn't that neat? But guess what? There are other aerobic respirators, such as yeast, right? Have you ever heard that yeast are used to make beer and wine? Yeast are aerobic cellular respirators. However, if you deprive them of oxygen, what happens? If you deprive them of oxygen, they switch to fermentation, which means they're only doing glycolysis, which means they're only making 2 net ATP. But did you know their fermentation product is different? You and I make lactic acid. However, these creatures, during fermentation, they make the byproduct ethanol, drinking alcohol. Isn't that neat? So all I want you to know is that A, what causes fermentation? Remember, lack of oxygen. Um, but also that different creatures can give different fermentation products. Like you and I, we produce lactic acid for our fermentation product. Yeast would produce ethanol, and there's even other fermentation products out there too. There's a bunch of different creatures that make different fermentation products during fermentations. Just be aware of that. So that leads us to clicker question number 5. Let's try this together. What is the main advantage of fermentation over aerobic cellular respiration? A. Fermentation generates ATP even if O2 is not present. Isn't that the answer? I think that's the answer A, right? Because fermentation can proceed even if oxygen is not present. So yeah, there you go. That was the whole answer right there. Pretty straightforward. I hope that made sense. So let's take a quick little break time with Gizmo and Wicket and see what these little guys are up to. And then we're just going to finish the last few slides strong. All right, welcome back from break time with Gizmo and Wicket. Let's go ahead and finish off this chapter strong. And I know it's been a doozy of a chapter, but thanks for bearing with me here. And now that we understand both photosynthesis from chapter 5 and cellular respiration from chapter 6, we can actually compare and contrast the two. And that's what this final part of the uh, chapter is all about, this final section, section 6.9. Now, take a look here. I want you to realize something. Do you remember that cellular respiration has certain inputs and outputs, right? What do you require for cellular respiration? Do you recall? Look, glucose is required for cellular respiration as well as oxygen, right? And what's produced by cellular respiration? Remember, H2O and CO2 as well as ATP. Okay. And now let's look at photosynthesis. I want you to look at this. Is so neat, everyone. Look at this. Remember what was required for photosynthesis? Remember, photosynthesis requires water and it requires CO2. Well, aren't water and CO2 provided by cellular respiration? Aren't those products of cellular respiration? So you need CO2, you need water and sunlight. And what are the products of photosynthesis? What does photosynthesis make? It makes glucose. So look at this. It's like a cycle. Look at this. The what I'm trying to say is that the waste products of photosynthesis are actually required for cellular respiration, and the waste products of cellular respiration are required for photosynthesis. Isn't that neat? In fact, these are called converse reactions. That means that the arrow is going in one direction, for cellular respiration, and photosynthesis essentially is the same reaction but backwards. It's the reverse reaction. Isn't that neat? And this is really interesting because I want to do a thought experiment with you. you know if you and I were put, if a human was put in a jar and then they put a plug in the jar, like a cork in the jar, right? We would soon breathe all the oxygen and we'd probably, perish, right? So we couldn't live in a corked off jar. Isn't that weird? Yeah. I mean, that makes sense. I mean, we would breathe all the oxygen in and we would convert it to CO2 and then, we wouldn't be able to continue breathing because there wouldn't be enough oxygen in the jar. But imagine now, instead of a person in a jar, we're a plant in a jar. Did you know that plant in the jar, as long as it has access to sunlight and it has, it's already started out with, it has soil and water already, but imagine if we plugged that jar with the plant. Did you know that that plant will not perish? It will not um, run out of oxygen, and it will not run out of CO2. You know why? Again, because of this slide here. The waste products of the mitochondria are required by the chloroplasts, and the waste products of the chloroplasts are required by the mitochondria. So you could, yes, you could definitely plug a jar with plant inside, and as long as there's sunlight, the plant will live and live and live. And there's actually a person who's done this. He's actually grown a plant in a closed off jar for over 50 years and it's still alive. Isn't that amazing? And now you know why, right? It's because these two organelles are kind of like yin and yang, right? They allow the plant to live on. The plant is a producer. It doesn't need to eat other organisms, and it can even breathe on its own because it produces its own oxygen and its own CO2. Isn't that neat? I think that's fascinating to understand. So with that, I believe we've reached the end of the chapter. I hope it made sense. Again, remember, I have that other more detailed cellular respiration video that I had linked to earlier. I hope you got a lot out of this. Let me know if you have any questions in the comment box below. But otherwise, I'll see you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.